Welcome back for the third and final part of our discussion on cells. So far we've talked about the cell membrane. Remember, that's what separates the cell from all of its surroundings. And then inside of the membrane, we've talked about the cytosol, we've talked about the organelles that float around in the cytosol. And so the last thing that we haven't talked about, obviously, is the nucleus. And so as you're probably familiar or probably remember from your early science classes, the nucleus of a cell contains its DNA. And remember that the thing that I want you to know about the DNA is that it is the instructions for building proteins. And in fact, one of the ways that this book refers to cells is as being miniature protein factories, because that's one of the primary things that cells do is to make proteins. And of course, in order to assemble them, to make them, they need the instructions on how to do that. Now that said, cells make other things, right? Cells can also assemble carbohydrates, and they can assemble lipids, which are fats, and then combinations of them, right? But the structure of those carbohydrates and of those lipids is not dictated by DNA. So DNA is the instructions for building proteins, and again, DNA resides inside of the nucleus. Now most cells only have one nucleus. Some have none, and we'll talk about that here in a second. And then other cells, a few cells in the body, are multinucleated. And so if you've already had structure and function 1, we talked about skeletal muscle cells, the fact that they are multinucleated. And in fact, that's actually part of the adaptation process to strength training, for example. So if you've had 201, this is a review. If not, it's a preview. Either way, it works. So if you engage in strength training and the muscle cells grow, right, so that's muscular hypertrophy, part of that process is the migration of satellite cells, which you can basically think of as a type of stem cell, and they actually fuse with the skeletal muscle cell, and their nucleus then becomes part of that skeletal muscle cell. So we add nuclei to those skeletal muscle cells as part of the adaptation process in response to strength training. And so one of the things I talked about in 201 is the idea that obviously all the adaptations to exercise work under this principle of use it or lose it. So if you lift weights for 3 months and you gain some muscle mass and then you stop lifting for a little while, you're going to lose that muscle mass. But one of the things that happens is even though your individual skeletal muscle cells shrink in size, they maintain those extra nuclei that they got as part of the original adaptation process. So when you retrain again, so after you lay off for a few months and then you start lifting weights again, the time that it takes in order for you to recover the muscle mass that you lost is going to be shorter because the cells are already have those additional nuclei from the initial adaptation. So why that matters then is the more nuclei a cell has, probably the more proteins it produces, but in skeletal muscle cells, the more nuclei they have, the more quickly they can produce those proteins and the more quickly they can recover those lost training adaptations. So I mentioned that some cells have no nucleus, so the key example there is red blood cells. So red blood cells, their job, as we'll talk about later, is to transport oxygen. And so if red blood cells had a nucleus, well, that's going to take up a bunch of space inside of the cell, and that's less oxygen that individual red blood cell can carry. So mature red blood cells don't have a nucleus. But as a result of that, because they don't have a nucleus, well, they don't have the instructions to make proteins, which means that they don't have the instructions to repair themselves. So red blood cells then have a fairly short lifespan. It's somewhere in the neighborhood of 3 months because they sort of accumulate this damage from squeezing through small blood vessels, and so you start to get damage to the cytoskeleton. But because they lack the instructions to build the proteins to repair the cytoskeleton, they can't make any repairs, and so ultimately those cells fall apart and effectively die within the course of several months. So a lack of a nucleus means a cell can't repair itself. Cells that have lots of nuclei or multiple nuclei are really good at making proteins and making lots of proteins. All right, so in terms of the regions of the nucleus, so you can see a nucleus pictured there, and you can also see that the nucleus is continuous with the rough endoplasmic reticulum. So that's all this blue stuff here. Remember, the rough endoplasmic reticulum is rough because there are ribosomes studded in it or embedded in it. And also remember that ribosomes are technically organelles, but ribosomes are responsible for assembling proteins. And we'll talk about ribosomes quite a bit today. So as you can see there, the nuclear envelope has two membranes to it, or it's a double membrane. So that outer membrane of the nuclear envelope is continuous with that rough endoplasmic reticulum. And so we're actually going to assemble some proteins from the cytosol into that rough endoplasmic reticulum through some of the pores that I'll show you here in a little bit. So the outer portion of the nuclear envelope then is continuous with the rough ER. The inner portion of the nuclear envelope, or the inner portion of the membrane, is responsible for effectively serving as a scaffold for the DNA. So your cells contain lots and lots of DNA. The estimate is if you stretched it all the way out, it would be about 2 meters worth per cell. And obviously cells are microscopic, right? And so you have to condense quite a bit of genetic information in a really small space. And so we consolidate DNA down, and I'll show you how we do that here in a second, into chromatin. But we consolidate that down so that it fits inside of the nucleus, and so that inner membrane serves as a sort of scaffolding to enable us to kind of consolidate that genetic information down. The nucleoli, nucleoli is plural, nucleolus, as you can see here, is singular. So most nuclei have 1 or 2 nucleoli. The only thing I want you to know about the nucleoli, so they're basically these darker staining areas. Like I said, there's usually 1 or 2 inside of a nucleus, and that's where we assemble the ribosomes. So when I talked about ribosomes, I don't think I mentioned last time, so ribosomes have 2 subunits. There's a large one and a small one, and I'll show you pictures of what that looks like here in a second. But those 2 subunits are assembled inside of the nucleoli, and then the ribosomes migrate out of the nucleus through some of these pores. Other things, so chromatin, as mentioned, is the way that we kind of consolidate the DNA, the way that we sort of consolidate lots and lots of information in a relatively small space. So chromatin consists of DNA, but in addition to DNA, because DNA actually only accounts for about 30% of it, about 2/3 of it, or a little less than 2/3 of chromatin, is something called histone proteins, which is down here at the bottom. And so histone proteins play a couple roles. So here's some DNA, and here's some chromatin. I'll show you a little closer up. So here's DNA, and it's double helix, right? And you're familiar with that shape. These little discs here, as you can see, are histones or histone proteins. And so the way that we package DNA to make it take up less space is in the form of something called a nucleosome. So nucleosome is the subunit, if you will, of chromatin. And so a nucleosome includes these 8 little histone proteins, which again look like flat discs, and then the DNA coils around that twice and then moves on to the next nucleosome, so the next batch of 8 little histone proteins that it'll wrap around twice. So again, helps us consolidate lots of information in a relatively small space. The other thing that histone proteins do, in addition to helping us package DNA, is that they lost my train of thought there. The other thing they do is that they sort of either allow or inhibit access to the DNA. So you've got over 25,000 protein coding genes in your DNA. So we make lots and lots of different kinds of proteins, right? We've got lots of instructions to do that, but we're not making all of those proteins all the time. And so when we don't need that particular set of instructions, we sort of store it, again, by wrapping it around those histone proteins. And then the histone proteins determine which genes, which instruction sets are available and which are not. So they basically can act to, not necessarily turn off genes, but they act to limit access to certain genes, which again are sets of instructions to build proteins. All right, so that's DNA. So again, DNA is the master blueprint. It's the instructions for building all of our more than 25,000 proteins. But in order to actually assemble those proteins, we need RNA. And so there's 3 types of RNA that you can see down there at the bottom of the screen. But probably the easiest way to conceptualize RNA is as the assembler, as the builder of proteins. So a few terms on here. So DNA, I showed you on the previous slide, you're familiar with the double helix shape of DNA. DNA is comprised of a backbone and then a series of nucleotide bases. And so the 4 nucleotides, you're going to see lots of just the initial letter, but you've got adenine, thymine, guanine, and cytosine. And so they are always paired in the same way. So adenine is always paired with thymine. Guanine is always paired with cytosine on the DNA. So I'm going to click over one more slide and then I'll come back. So for example, you can see here down at the bottom, so adenine are these little green pointy ones, thymine are the yellow pointy ones, and so we've got an adenine here on the DNA. We're going to ignore, well actually let me back up, let's go over here. Let's make life easier. So here's your DNA double helix here. Here are your nucleotides. And then the complementary ones are on the other strand of DNA. So if you've got our adenine here, the little green arrow, its complementary nucleotide is thymine, and so that'll be on the other strand of DNA. And then if we've got our cytosine here, then its complementary nucleotide is guanine, which is here. And then we've got another cytosine down here. Complementary nucleotide is guanine here. And so you've got this series of repeating ATGC in whatever order. Those are the nucleotides. And so there are 2 strands on DNA. There's a coding strand and a template strand. We'll talk about those here more in a minute. But effectively, the A and T are going to be interacting with each other on the complementary strands. G and C interact with each other on complementary strands as well. So they're always paired like that. All right, some other terminology. So amino acids are the building blocks for proteins. So proteins, the more technical term for them, which this book uses quite a bit, are polypeptide chains. But proteins are effectively just long strings of amino acids. So for example, a relatively small protein consists of a string of 70 amino acids. So you've got 70 amino acids all linked together, and then that forms a protein. And obviously proteins can be much, much larger than that, but as I mentioned, that's a relatively small one. Other terminology, so triplets and genes. So each series of 3 nucleotide bases is a triplet. So a triplet is a series of 3 nucleotide bases. And each triplet codes for an amino acid. So each series of 3 nucleotide bases codes for an amino acid. So for example, sticking with this, right? So we've got an adenine, a thymine, and then a guanine. So we've got ATG in order. All right, and so then if we click over to, this is table 3.31 in the book. So what did we say we had there? We had ATG. All right, so our first letter is A, which then becomes a U on RNA, and we'll talk about that in a minute. And then G is the third one, so that makes it AUG. So that then codes for the amino acid methionine, right? So each series of 3, each triplet codes for a specific amino acid. So on this table on the right, those are all of your amino acids. And so you can see the different series of triplets. So for example, if we have, let's go with this one here. So if you've got a C, and an A, so cytosine, cytosine, and then adenine, sorry, cytosine, cytosine, and adenine. I was going to say arginine, and I knew that was wrong. So these 3, so that then codes for PRO, which is the abbreviation for proline. So that's the amino acid that's coded for there. So each 3 of these in a row, each triplet codes for a specific amino acid, right? And so then if we got one amino acid here, we got the next 3 code for another amino acid, next 3 code for another amino acid, etc. So that example that I gave you a minute ago of a small protein consisting of 70 amino acids, well, if we multiply that out by 3, because you got 3 nucleotides that code for each amino acid, you have a series of 210 nucleotides that have to code for that, that protein. All right. So triplets and just to make life confusing, there's another term which means the same thing. So triplets and codons are the same thing. So they're both a series of 3 nucleotides. So triplets, series of 3 nucleotides. Each triplet codes for an amino acid. Each amino acid then builds proteins. Okay, so that's kind of the way that it runs. Genes are the instructions for building proteins. So the gene for building that protein that consisted of 70 amino acids, the gene would be that series of 210 nucleotides. So the genes are the instructions for building an individual protein. They're comprised of a series of nucleotides, which are divided into triplets, which then code for amino acids, and then we build our protein. So one of the things I want you to know then is a gene is an instruction for building proteins. A couple more terms on here, exons and introns. So once we have, and this is on both DNA and RNA, so DNA has extra information in it, if you will. So there's information that actually codes for a protein, and then there's sort of extraneous information that at one time was thought of as sort of genetic junk, but it's now thought that that's more like a scrapyard of nucleotides that we can pull from. So exons then are the informational sequences that actually code for proteins, as opposed to introns. Those are non-coding, so they don't code for specific proteins. Sorry. So exons code for proteins, introns don't. Introns are basically extra nucleotides. So when we're making proteins, one of the things that we'll do is actually to trim off the introns, to trim off the extras as part of that process. All right, in terms of RNA, so again, a way to conceptualize RNA is as the builder of proteins. So there's 3 types of RNA. So there's messenger RNA, and the abbreviation there is lowercase m RNA. So messenger RNA's job is to basically write down the instructions for building a protein. So messenger RNA, one of the other things to know about it and why it's necessary is that DNA can't leave the nucleus. So DNA is stuck in the nucleus, but we assemble our proteins by and large in the cytosol of the cell. Well, if we're going to assemble it, the protein's in the cytosol, but the instructions are in the nucleus, we have to get some version of the instructions outside of the nucleus and out into the cytosol. And so it's the job of messenger RNA to do that. So I mentioned earlier that on DNA, the base pairs are A and T, G and C. Well, again, to make life complicated, messenger RNA doesn't have T on it. It instead has a U. So if the DNA base nucleotide is an A, the corresponding pair on messenger RNA is going to be a U. So the T becomes a U anywhere on the mRNA. So just something to be aware of. And then the ribosomal RNA is the same thing as a ribosome. So the ribosomes, again, are going to actually assemble the proteins. And then the transfer RNA is out in the cytosol. The transfer RNA is kind of like the gopher in that it goes and gets amino acids in this case. So it's sort of the errand RNA, if you will. So it goes out and gets the building blocks. So transfer RNA gathers the building blocks. It's the gopher. So those 3 work together. So the messenger RNA has a copy of the instructions. Ribosomal RNA reads the instructions in concert with transfer RNA, but it reads the instructions, actually assembles the proteins, and then transfer RNA goes and gets the building blocks. So let's get into the specifics of that. So there's a couple of graphics I already showed you. And I left this, it's an animation, I left this in the slideshow that's up in Canvas. So if you want to look at the animation, it's obviously a cheesy textbook animation, but it does a pretty good job of explaining the relationship between DNA and RNA. All right, so there's 2 processes here. There's transcription and translation. If we transcribe something, we write it down. So think of something like a court transcript is the first one I can think of offhand. So a court transcript is a record of court proceedings. So, if you get a speeding ticket and go contest them, there will be a transcript of those court proceedings, right? And so transcription is just writing it down. And so in addition to writing it down or copying the message, because we're changing the message from DNA to RNA, we're actually changing the formats. So the book kind of talks about changing from one file to another. They talk about audio files, but you can think of it as changing the format from a Word document to a PDF document. The information is still the same, but the presentation is a little bit different. So that's DNA transcription. So again, from a practical standpoint, what's happening here? In order to build a protein out in the cytosol, I got to get the instructions from DNA and get them out to the cytosol. So messenger RNA is going to do that. But first, it has to write them down. And so the process here, so this purple stuff, that's our DNA, right? And so we have pulled it off of those histone proteins. So we've exposed that DNA. And then we have an enzyme called RNA polymerase that is then going to scan that DNA line until it finds this promoter region. And once RNA polymerase finds that promoter region, it's then going to attach to that promoter region, and then it's going to split those two strands of DNA. So it's going to break the bonds between the A and T and G and C, right? And so what you see down here in the lower right, the larger picture, is that now we've got our RNA polymerase here. It has split those two strands of DNA. And as mentioned, those two strands, the top strand here is the coding strand. The bottom strand down here is the template strand. All right. And so then mRNA comes in, and it's then going to assemble the complementary nucleotides. So remember that on DNA, and I'm going to stick with the colors that the book uses just to make life a little bit easier. So what you've got here, this little yellow one, so that is thymine, okay? And so then thymine's complementary pair, or complementary nucleotide, is adenine. And so you've got our first thing on our first nucleotide on our mRNA is going to be our adenine. And then the blue one is cytosine, and so its complementary pair is guanine. And then we've got adenine here. And so normally on the other DNA strand, on the coding strand, this would be a thymine, because if A is down here, then this should be T. But remember that on RNA, the T's get replaced with U's. So this is actually a U right there. So that yellow one becomes a U. And then this red one is guanine. So we got a cytosine. This yellow one, because we're still on the DNA strand, is thymine. It gets replaced by adenine, etc. So then we just start replacing all, or not replacing, but we start assembling all of those complementary nucleotides. And then we assemble them into a string. We run all the way down to this gene. So all of this from the promoter region to this tan area is the termination signal. So everything from the promoter region to the termination signal, that is one gene. So that is one set of instructions for building one protein. And so the RNA polymerase will run along this gene, copying down the information on, I say copying. So it's going to assemble the complementary pairs to the template strand. So one of the things to understand is that the mRNA here is going to be an exact copy of the coding strand. And it's going to be a set of complementary nucleotides to the template strand. So mRNA, exact copy of the coding strand, with the small caveat that the T's become U's. And mRNA then is the complementary nucleotides to the template strand. So then once RNA polymerase gets all the way here to the termination signal, it's going to detach. Now we have our completed messenger RNA that is a copy of one gene, a copy of a set of instructions to build one particular protein. And again, the mRNA is going to be a copy of the coding strand. All right, and so then our mRNA is going to move outside of the nucleus. And a couple more terms on here. So we talked about codons. So remember that codons are triplets. So they are more specifically a triplet on messenger RNA. Transfer RNA, remember I mentioned that transfer RNA is out in the cytosol, and its job is to act as the gopher. It goes for amino acids. And so it goes and gets the building blocks of the protein. And so the transfer RNA has an anticodon on it. So messenger RNA has the codon, right? And so clicking back to this slide, so these three here are a codon, And again, they're the copy of these 3 on the coding strand. And so then transfer RNA goes and has an anticodon, which is a copy of the template strand. Okay? And so transfer RNA goes and gets the particular amino acids that correspond to this particular anticodon. So this anticodon is UAU. And so if I click back to our handy little table here, so the first letter is a U, the second letter is a U, and then the last letter is also a U. And so, oh wait, whoops, sorry, UAU. I should probably get it right. All right, so first letter is a U, second letter is an A, and then third letter is a U. So our UAU here codes for tyrosine. Okay, so TYR, and then that is tyrosine. So that particular amino acid is tyrosine. So this transfer RNA is only going to go and attach to tyrosine. Okay, so the transfer RNA has anticodons, which are a nucleotide sequence that corresponds to particular amino acids. All right, so once our messenger RNA is out here in the cytosol, transfer RNA in the cytosol with the code for methionine, which is an amino acid that corresponds with the start of a gene, the start of a protein coding sequence. So we've got these transfer RNAs out here that have methionine on them, and so when they see messenger RNA, they're going to attach to them along with the small subunit of a ribosome. So remember I mentioned a minute ago that ribosomes have 2 subunits. So there's the small subunit here and the large subunit here. And usually the way ribosomes are described is looking like an acorn with a cap on it. So the small subunit's obviously the cap, and then the rest of it is the rest of the acorn. And so our transfer RNA with methionine is going to attach to our messenger RNA along with the small ribosomal subunit. And then once they kind of scan up and down the messenger RNA until they find the start signal. Once they find that start signal, then the large subunit of ribosomal RNA, or the large subunit of the ribosome, attaches. And now we're actually ready to assemble our protein. So we've got our initiator, transfer RNA, mRNA, and then the small subunit. And so now, once we've found that start signal, now we're ready to start assembling a protein. So that's the initiation. Step 2, or stage 2, elongation, is effectively assembling the protein. So the large subunit of the ribosome has 3 areas, if you will, to it. So there's an A region, a P region, and an E region. So what those correspond to, effectively what's going to happen is that your ribosome is going to move from left to right. In this depiction, it's going to move from left to right on your messenger RNA to assemble this protein. So the A region is where this transfer RNA is, so transfer RNA is going to recognize the code for this, which is, so CUA, okay? So CUA is our codon, and so now we need the anticodon that corresponds to that. So C, the anticodon is going to be G, U, the anticodon is going to be A, and then A, the anticodon is going to be U. So the anticodon here is GAU. And so if we go back to our table back here, so we're looking for a G, an A, and a U. And so we're looking for ASP, which is aspartic acid. Okay, so that little code there codes for, oops, click over one. That little code there then codes for aspartic acid. And so we're going to get that coming in. All right, so there you go. So then the transfer RNA will come in, it'll attach, and then once we've got the next amino acid here attached, the ribosome is going to slide over one, and when it does that, when it slides into this T-spot, we'll add this next amino acid to the one that's already there. So we'll add that to that proline that's already there. And then the ribosome will slide over one more, and then that transfer RNA will be ejected. So in the E region, you get ejection, gets kicked out, right? And so we're just assembling additional amino acids here. Okay? And then once we get to, so you've got codon recognition, which happens in the A region, says, hey, go get this particular amino acid. Then once we've got the amino acid in the P region, we attach that to our string of amino acids we've got going on already. And then in the E region, or translocation, we're going to get rid of that. We're going to get rid of that transfer RNA. And so we string them along, or we assemble these long strings of amino acids, and then eventually that makes a protein. And then once we hit that stop codon, or once the ribosome hits the stop codon, you get separation of the large and small subunits. So the ribosome splits in half. We add water to our polypeptide chain, so to our protein, to free it up, and then it floats off to go do its job. So oftentimes, remember in our rough ER, our rough endoplasmic reticulum that is continuous with the nucleus, you're going to have ribosomes studded into it, where these pores are. And so we're actually going to read and assemble the proteins into, the string of amino acids is actually going to go into that endoplasmic reticulum. And so you can see, here's our budding protein here, and then we snip it off. Inside of the endoplasmic reticulum, we can do things like add sugar or phosphate groups, and then remember the endoplasmic reticulum will package that protein and then send it off to, oftentimes, to the Golgi apparatus, where it'll be modified, and then it'll go wherever else it needs to go. All right. And so this is just another, I thought it was a helpful graphic here because it includes the triplets, the codons, and the anticodons, right? And so the reason I think it's helpful, so remember that you've got your the different strands of DNA. So you've got your coding strand, your template strand, etc. Not etc. Those are the only two. Sorry. So anyway, so this up here, the top one would be your coding strand. And so then the anticodon is going to be the same thing as the coding strand, except that, again, bring my mouse back, the T's and the U's get swapped out. So coding strand, anticodon, those are the same. And then the coding strand and the mRNA are going to be, we'll leave it at that. All right, so then you've got, so here's your first gene, is the purple one here, which is what's being copied. Second gene is the yellow one here, and then on down, right? So yeah, so I think it's a handy little graphic, and that's it for cells. So that's the end of the cell talk, and then next time we will start on tissue.